Thank you very much for the introduction and for the possibility to talk. I think I will talk from here because I need to switch the slides. So let me talk about what we are doing at Gero and what we have learned about aging with our friends at Gero and what we think we should be doing to actually produce a meaningful intervention against aging. We have just had a panel, which I wish we could do in the other way. So first tell what we are doing and then discuss. Well, aging, there are multiple definitions to aging. For me, the most, let's say, interesting one is the exponential amplification of your chances to die of all causes as we age. So for fun, because normally people do log plots, like you have these straight lines, for fun I just plotted the probability of death in humans as a function of age and natural coordinates, just for you to understand how bad the problem is. So the chances to die of all causes double every 8 years in humans. This counts also for death from accidents, for example. So, a busy street is not dangerous for a teenager, but it's dangerous for an elder individual because of neurophysiological deficits. By the way, we have animals that presumably almost do not age. We have animals like naked mole rats, where the same graph, no log plot here, just the plain mortality as a function of age, stays more or less constant as a function of age for thousands of years. Of course, doesn't prove that these guys do not age, but these guys are aging very slowly. And the comparison between these two graphs actually brought me into the field because I want to be like that guy, not like those two guys. So my background is physics, and whenever I see an exponential disintegration of something, I kind of know by heart from textbooks that this is exponential instability. So our first idea was that there should be kind of two kinds of animals. There should be animals that amplify errors, and there should be animals that presumably do not. So basically we started thinking that all living beings could be classified into two situations just by observation, right? Because we have those naked mole rats and we have the humans or mice that are exponential agers. We were thinking what would happen if you produce any kind of error into a biological system. For example, I take out a molecule out of an organism. So that molecule was there for a reason, most probably it was doing some job, so now it's not doing any job, which means that it will produce or not produce other molecules and that there will be kind of expanding hole in the whole organism of missing or extra molecules. And then in some species, this disturbance can be cleaned out. So if repair systems are working very well, then on average you would not, even if you introduce an error into the system, you wouldn't produce more errors on average. Or there could be systems where one error amplifies errors and those amplify more errors, and then you start exponentially disintegrating. So the closest analogy here is the infection. You could have a situation where the infection is controlled, when you have quarantine, immune system, and everything else. If you put a virus into the system, then the system just, eats it and nothing happens. Or you could have a situation where the virus produces more viruses and so on. So we can talk about error amplification coefficients. If the system is unstable, error amplification coefficient is larger than 1, you disintegrate exponentially. If it's below 1, you just leave, okay, not forever, but at least you don't have aging. So we decided to sort these things out using lots of data. So this is a fully data-driven approach. It's like weather prediction or market predictions. As I said, I'm coming from physical and engineering sciences, which means that all these arguments can be actually backed up by calculations. So we started from mice, because when we started this, mice were kind of most studied animals on Earth. But things changed, and over, let's say, 5 years, I firmly believe that humans are now the most studied animal on Earth. We have tens or even hundreds of millions electronic medical records in digitalized form. We have tens of millions of genomes in humans. We have lots of molecular data. So in fact, we are now in a good position. We can study aging and diseases in humans first. So this approach, I would say, gave some fruits. We published a bunch of papers. We got a collaboration with Pfizer precisely on this. Pharma is actually very interested in what people are doing in the aging field. They are interested, they are not impressed, I would say. The problem with pharma is that in terms of effect size and our bravest aspirations, it's still a little bit below what they expect from a drug. But that's very interesting, because I will tell you in a moment how these kind of models can actually inform pharma how to separate aging from diseases, how to help pharma make drugs against aging, against diseases, but not aging. For the God's sake, not make a drug against aging, and at the same time study aging. So, as I said, we use the data, and we are using longitudinal data. Longitudinal data means that you study the same animal multiple times. This is very different from most of the work that is done in biology, where you have huge biobanks and you have just one individual measured only once. You don't know how this person ended up here at this point. So you have the outcomes, but you don't know the history. Even if you have long-term outcomes like 30 years survival, this is cool, but this is not sufficient, because you don't know how the phenotypic molecular changes are translated in mortality and diseases. So this is just one graph in mice. In mice, we were actually able to put this theory in practice. We actually measured the error amplification coefficient in mice. We proved that if you have a disturbance of, if you have two mice, a mouse A and mouse B, which are different by some parameters, you can actually see in the data that the distance between these two mice in parameter space will get exponentially larger. So two animals become less and less similar to each other. So all errors are exponentially amplified in mice. So this is the actual graph from one of our papers. So this thing that we can call the total damage accumulated so far, or the biological age, is actually an exponential function of age. And I like on this graph those red dots. The red dots are the biological age measurements of mice that are scheduled to be killed in the laboratory because they are too sick. You know, in a good laboratory, you kill animals because they are sick, so they are not allowed to die from old age. And you can see that the animals are sacrificed when they reach kind of ceiling in biological age, right? So in mice, things are very simple. The biological age shoots up exponentially, the system is unstable, and when it hits a certain level of toxic damage, the animal is dead. So this is really this exponential amplification. So it sounds scary. I mean, exponential amplification of errors as a cause of death, this is scary. But it also has a very interesting consequence. If your system is unstable, it doesn't know what is the equilibrium position. There is no equilibrium position. There is no homeostatic equilibrium position in mice. Which means that if you find an intervention that reduces biological age at a certain point in time, the system wouldn't know how to get back to the control. The system will stay younger forever. And that is what we see in infinite number of experiments in mice in different groups. Just in our hands, it works like this. So on the vertical axis, I have the biological age. On the horizontal axis, I have time. And then here, in between the first 2 points, I apply only once the anti-aging intervention, right? So just one shot of a drug. And then you can see that since the system is unstable, this is the precise definition of instability. Since the system is unstable, it never gets back to the control. So the mice are younger forever. So I would tell that if you have any intervention that acts against aging in mice, if you do it once, the mice will remember that forever. It's very easy to rejuvenate a mouse. And I think we have lots of evidence that this is happening. Just maybe to show that these are not only computations, this is just one of our examples, which is already particularly old. We worked with Brian Kennedy at the National University of Singapore. These are survival curves. On the vertical axis, you have the number of animals alive to a certain point, and then along the horizontal axis, you have the time since the beginning of the experiment. These are geriatric mice, so they are 100 weeks old at the beginning of the experiment. You can see that in the control, they are dying of aging. So you can see how these guys are dying of aging. And then we have one shot of experimental intervention that was predicted by our calculation. And you can see that even though there was just one intervention, remember the instability, there is a mortality delay. So these guys are younger. And 2 months after the treatment, you can measure essentially any hallmark of aging there. For example, the amount of senescent cells in the immune system of these guys. And you would find that there is no drug in the circulation anymore. But these guys are younger. We used the same approach in humans, and we were totally surprised. I mean, that's not something that I was expecting, because I started with the fact that humans are exponential agers, so our chances to die actually go up exponentially as we age. So I was expecting that if I built a system that would predict a biological age in humans, I would find that this biological age would also go up exponentially, as it does in mice. It does not. Of course, I don't have enough data to plot the biological age trajectory along the whole trajectory of life in humans, but schematically it looks like this. All humans are born in a state where there is a solid homeostatic equilibrium, which means that your biological age oscillates all the time. That's what Vadim, by the way, is showing, that if you apply a stress to a biological system, the biological age is going up and down, and then it tries to recover. In mice, it never recovers at the end, but in humans, it totally recovers. I think that's what we know from experience. If you are young, if you go to a party on Friday, you pretend to be working on Monday, and that's okay. Elder people also want to go to party, but they cannot pretend to work on Monday because it takes longer time for them to recover from the intervention. So aging in humans is not a matter of level of certain things like biological age. The biological age is kind of irrelevant in humans because the biological age oscillates all the time. You didn't sleep enough, the biological age is going up. You eat too much, it's going up. Your exercise is going down. But it's getting back to the equilibrium position. What happens, and for those of us who care to measure their weight along our life trajectory, we know that once in a while we have kind of earthquakes in our bodies when the equilibrium position changes. So once it happened, your equilibrium weight is now a different thing, and no matter how many exercises you do, you would never get back to this. I mean, you can reduce your weight, but when you stop exercising, it will go back to a new level. So what happens is that, you can never see it in cross-sectional data sets where there is only one measurement. If you have longitudinal data, and we have lots of that right now, you would see that instead of those smooth trajectories like, all the biology papers are telling you about, in humans you have fluctuations, jump, fluctuations, jump, fluctuations, and then on average you have those smooth curves. So what happens with humans is that we have those jumps, and very late in life, we experience a very large jump that produces the instability of the biological age. That becomes, frailty and accumulation of chronic diseases. So in order to prove that these things do occur like this, we concentrated on measures of aging that are not the measures of levels of certain things like biological age. We studied the fluctuations of biological age from blood analysis, from wearable devices, from methylation, doesn't matter, from anything. So what we observed is that when you have sufficiently long trajectories, you can actually, as I said, your life is a source of perturbations. It always perturbs you in such a way that your biological age is fluctuating. So you wouldn't call it even a biological age if it's fluctuating, right? It's more like a measure of stress. It's really the measure of quality of your lifestyle. Your biological age is going up when you're living toxic lifestyle, it's going down if you're living a healthy lifestyle. The challenge here is that if you stop doing what you have been doing, it's going back to the same level, right? So you don't care. So the interesting part is that if you measure how quickly you go back to the equilibrium level, you will find that the elder you are, on average, the longer it takes to get back to the equilibrium level. So on the vertical axis is the inverse time, the rate at which you approach the equilibrium level. The horizontal line is the age, and you can see that your resilience, your ability to come back to the norm, to recover from perturbations, is going down to zero and would disappear at about 100 years old. So this graph essentially is telling you that if you're lucky, you don't have lots of diseases and you survive till 100 years old, your resilience is so small that even a minor stress event will kill you. So this is actually aging in humans. So interestingly, that contrary to aging in mice, aging in humans is not related to biological age. It's related to a dynamic property of the biological age. Most importantly, the recovery rate of the biological age. And that's not just a mathematical exercise. So on the graph on the right, I plotted the same. I plotted the inverse hospital stay as a function of age, right? This is, you have a life-threatening event, so they put you into the hospital. It's an inhuman way of measuring human resilience, right? This is too much of a hassle to do it at scale, but if you have a large biobank, you can plot inverse hospital stay as a function of age, and you could see that exactly as we see in biomarkers, this thing is going down linearly with age and approaches zero, which means that the recovery, the hospital stay is infinite, you are dead, at about 120 years old. So with that, I want to conclude that in humans, contrary to mice, aging is not a property of the biological age levels. This only happens when you are very old. Aging in humans is a property of fluctuations of the biological age level. And the human life history, I can schematically, this is a schematic representation, but all the parameters of these visualizations are actually computed from the data. I can visualize like this. So when you are born, okay, let this guy die. Okay, he's dead. Now we have a new human being. So you can see that the organism state is stable. We are born into a stable state. So life is hitting us, perturbing us, but we are getting back. But then as we age, our recovery potential disappears, and then a small shock throws us away. And then our biological age becomes exponential, like in mice. Then we hit the wall, and we're dead. So once again, this is not just a metaphor, because we have computed all the parameters of these things. And this tells you that, once again, biological age is not relevant variable in humans for aging, but the parameters of these regulatory interactions are the change of the shape of that regulatory interaction is actually aging. So now you will tell me, where are my biological clocks? What are you doing? What you are doing so different from what other people are doing? And as I said, you have biological age and you have some other aging in humans. And this is actually what you can see in any kind of biological signal. You can take DNA methylation, you can take metabolomics, proteomics, doesn't matter. So if you do clustering analysis of trajectories of biological variables in humans, you would find that in any aging dataset in humans, there would be one feature that is exactly linear with age, and then it increases variance. This is a hallmark of a stochastic process, by the way. And then there are multiple features that depend on age in a non-linear way. And one of them would go to infinity at exactly 120 years old. So this is the weakest link. So this is the feature that is disintegrating first. So there are two kinds of aging clocks, and we know that. So there are aging clocks that predict chronological age, and there are aging clocks that predict mortality. And you can produce infinitely many other biological clocks that produce, risks of specific diseases. The most important part for us that there is one that is linear, and all the others are actually driven by the linear biological age, because they produce these nice quadratic curves, which means that there is a small nonlinear coupling to the first one. The first one is aging in humans. The first one is the thing that is changing the regulatory interactions and produces death. And the worst thing about this aging in humans is that this is not a single biological process. All the other guys are single biological processes, and this one is not. So this thing, the aging in humans, the linear feature in humans, is a result of uncorrelated bad things that happen to you over life. Like I think Vadim is telling, this is the damage. The damage is through nonlinear coupling, produces change into processes. All the other things are processes, and this is the damage. Why is the damage? Because it's linearly going up with time, so this is just uncorrelated events that get accumulated. And why it's damage? Because it's stochastic, that's why the variance is going up exactly linearly with time. So this is a Poissonian stochastic noise, for those who care. So this means that we are now in a very interesting, I would say, situation. Most of the things that we have learned in practice about aging is from mice. If you're a mouse, you are a happy thing because we can rejuvenate you at least once, or maybe a few times. I think Vadim can do it. We'll soon do it a few times in a row, right? We can make kind of a ladder of the biological age reduction in mice nicely. Once again, short interventions produces lifelong rejuvenation. Wonderful. The problem with that is that this phenotype is not relevant for humans. It's relevant only in the last 10 years of life, once you lose the dynamic stability. Of course, you can apply drugs late in life. They will produce effects. Just 1 week ago, I saw another results of clinical trials of anti-aging drug in humans, and everywhere this is the same. In humans, if you do it for old people, there is a large effect. If you do it in young people or relatively young people, the effect is small. Once you stop the treatment, the effect disappears. Right? So which means that there are two kinds of aging, there are two kinds of anti-aging intervention. One of them works in mice, rejuvenates a mouse, works late in life in humans, will produce infinitely many billions of dollars in sales, because it will produce improvement in quality of life of very old people. But this thing will not affect your cognitive decline, if you don't have Alzheimer. This thing will not affect your VO2 max decline, so this thing does not change this linear aging that occurs all the time in humans. So most of the industry is now working on this, we call this phenotype frailty associated phenotype. Most of the industry is working here. Why? Because we're testing our drugs in mice. And then there is another kind of aging, which is the stochastic damage accumulation, which also occurs in mice. But mice are dead within so short time that it doesn't matter. I mean, it doesn't have time to contribute to their lifespan, which means that the drugs that affect this true aging phenotype would not have large effect in mice. They will have some small effect, but not large effect. So with that, I would like to make maybe a few prospective statements, like we know that if we do experiments in mice, most hallmarks of aging in mice are correlated. If you have a drug that acts against any age-related feature in mice, it reduces all other age-related features in mice as well. It's very easy to produce lasting effects. There will be infinitely many papers about rejuvenation in mice. The very same drugs, and I think this will be unfolding in a few years to come. We will get the clinical trials. Now we have alpha-ketoglutarate. There will be, I know, other papers published soon. We will see that the same drugs that rejuvenate mice do not rejuvenate humans. They produce small, transient effects. You stop the treatment, the effect disappears. Which means that if we want to do drugs that produce multiple-fold improvement in human lifespan, we should be doing something else. And once again, I would like to refer to Vadim, since he came here. Vadim made all those signatures of aging in animals, of interventions, and across different animals, like mammals with different lifespan. I think the message from that slide is the same. The things that we're doing with interventions that are kind of primed to mice studies are not the same that are required to produce multiple-fold extension of human lifespan. These are two different worlds of interventions. And, well, I think there is a practical way to approach that. So this is just for intimidation purposes. So how to actually approach this? What to do about that? So to solve this problem, we need first to study aging not in mice, but in humans. Probably we can study it in other animals, but we don't know what kind of animals to study. So to understand how aging works in other animals, it's a lifelong project. So I think we should better study aging in humans just for practical consideration. So what we do is that we take very large data sets of electronic medical records. We're working with a data set of 10 million electronic medical records right now. You can get 100 million easily. There is another 60 million in the UK, so I think you can actually beef up these calculations by a lot. So what we do is that we take electronic medical records and we use machine learning in order to cluster diseases into a cluster of diseases with similar biology. And we look at the electronic medical records longitudinally along the life history of patients, and we try from the dynamic properties of these trajectories to predict what we don't know. And what we don't know is the total amount of damage in the system. That is contributing into the probability of transitions between health and disease. So this system infers biological age automatically. And this biological age is the target variable for our studies. So how does it work? So we are feeding hundreds of diseases, tens of millions of electronic medical records into these systems, and the system outputs the biological age as a function of age. By the way, we don't feed, well, okay, forget about that. We don't feed biological age to chronological age. That's not a good idea. But also on top of that, the system tries to predict which systems in the body are broken, and those systems are associated with related diseases. So for example, on the right graph, you see the probability of certain systems which are predicted by machine learning broken, and one of them is associated with cardiometabolic health. And this is, by the way, the probability of failure in this system is affected by biological age, and it's exponential and doubles every 8 years. So you can see that the system produces the estimate for aging, the biological age, and it produces the estimates for the health of subsystems that are related to diseases. And I think the system recovers what actually the medicine knows without GeroSense. So medicine has been knowing forever that we have a weak link in our body. This is a cardiovascular health. This disintegrates first, so this is the weakest link, so that's why you have the highest occupation of that cluster. And this is age-related, because the damage consists of infinitely many parts, and those things are uncorrelated. Each of them is benign, unless they cause cancer, but that's another story. And they accumulate over time, and they produce linear stress on systems and force them breaking. So the nice thing is that this system is totally unsupervised. We didn't ask the system to find cardiovascular diseases or other diseases. We only ask it to find the biological age and some clusters of diseases. And the system finds the biological age and diseases. So what you can do with this is this. You can now have, if you have 500,000 people with genetics, you have phenotypes, you have these clusters of diseases, you have biological age, and now you can start looking for genes that are controlling the rate of aging because you have the biological age, and you can find genes that are controlling risks of diseases. Okay, this is for demonstration, not for intimidation. We actually produced 3 genetic studies here, just to give you a flavor of what's going on. So here we have, on the bottom graph, we have lots of genetic variants in your body, and whenever you have a vertical line, it means that this genetic factor correlates to a disease. On the bottom line, we have the parental longevity GeroSense. As you may know, unfortunately, genotyping is, I'm kidding here, unfortunately, or fortunately, unfortunately. Genotyping is a young technology, which means that most people that contributed their genomes to biobanks are still alive, which means, unfortunately, that we cannot use them for genetic studies of longevity. But fortunately, or unfortunately, 2/3 of their parents are dead. And since some of the genes of parents are in the kids, you can still correlate genes in kids to the longevity of parents. So that's why it's called parental longevity. So you can see that there is a bunch of genes that are associated, vertical lines on the bottom graph that are associated with parental longevity. And then we do the same genetic associations with cardiometabolic cluster from the model and from the biological age. And you can see nicely that if you have any hit in parental longevity, almost all hits are either in cardiovascular health or in aging, but it's never like there is one hit in the cardiovascular health and aging. So it's nicely that the system separates genetic factors that are risk factors of accelerated aging and separately genetic factors of diseases. And this is the point where pharma starts liking you. Because pharma knows that aging is not reversible. I mean, technically, they need to do phase 2 clinical trial within a year or two, right? If you cannot reverse something within 2 years, it's irreversible, and they want to regress this out from the calculation. So we ended up ourselves in the business of regressing out aging for pharma. I mean, they, for the God's sake, don't make a drug against aging, because that would be too long to try, right? So the system outputs aging and disease risks. We regress out aging from our calculations. We help, we inform pharma about human evidence confirmed targets against diseases, and we have still aging as a phenotype that we can associate with genetic factors and everything else. So that brings us to the point where, I mean, first, we learned how to rejuvenate a mouse. We learned how to separate aging from diseases in the data. We learned how to partner on this knowledge from humans, by the way, not from animals, but from humans, with those guys who know how to do drugs. And still we have aging phenotype as a separate entity that we can study on our own and make experiments that are now squarely aimed only at search for factors that are controlling the rate of human aging. So with this I would conclude. Alex is here, we have a wonderful team, we have a bunch of people like Brian, like we have a number of publications with Vadim. We have Andrea Ulchenko, who is helping with the genetic studies. So we are looking for more brains and hands and computers, as usual, and data to make this all possible. I think that, as I said, the industry is, I believe, is on a very firm footing to solve the frailty phenotype. We have now dozens of companies entering clinical trials. We see the data from clinical trials. I mean, when we get old and sick, there will be a solution that would help us a little bit. I think now it's time to raise stakes substantially and go after the aging phenotype in humans that is not frailty, in order to help most of the people sitting here not to get to the point or get to the point where the frailty sets in as late as possible. So that's about it. Thank you very much. And if you have questions.